Hello everyone, Zia Hussain this side. I've done MSc Zoology, have qualified CSI and JRF with all India rank 65. I've qualified SET Maharashtra as well for the lectureship and one more exam that is called for the PhD, that is PET exam. Currently I'm doing my PhD at SPPU Pune. Our today's topic is the formation of the mRNA molecule, that is what we call as the transcription. So now let's start. Here, the process of formation of RNA molecule from DNA. The process of formation of RNA from the DNA, or specifically I have told you about the mRNA. So basically, during the transcription, the complete DNA do not forms the RNA, rather only one segment from the DNA forms the RNA. And one more thing, only one strand of the DNA participates in the transcription. Only one strand of the DNA participate in the transcription. Now the question arises, why? Because if both the strands get transcribed at the same time, so basically like the sequence of the amino acid will be different in both. Simple, like if the genetic codes are different, like the different amino acids will be there because they are complementary to each other. And one more thing, there will be two RNA molecules which will be formed, and both of the two RNA molecules will be complementary to each other. Both of the two RNA molecules will be complementary to each other. At the end of the day, they will be forming a double-stranded RNA molecule. So basically, like the transcription process will be failed. So that is the case why only one strand of the DNA takes part in the formation of the mRNA molecule. Hopefully it is clear with you. Now, if I am dealing with the word called as transcriptional unit, it basically has three regions. The first one is called as a promoter, the second one is called as a structural gene, and the third is called as a terminator. Promoter is structural gene and the terminator. So the first thing first, the promoter. It starts like the name suggests to you, it is just for the beginning of the transcription. A structural gene that is actually transcribed, that is the gene which is responsible for the formation of the protein. And the last but not the least one, it is a terminator which ends the transcription. So here promoter is beginning, structural gene is the actually transcription, and the terminator is the end of transcription. This is the case. Now, the two strands of the DNA, they are called as template strand, and the other strand is called as coding strand. The first one is called as a template strand, whereas the second one is called as a coding strand. Now, what is the name of the enzyme which is responsible in the transcription? It is called as RNA polymerase. Hopefully, you are remembering that the name of the enzyme which was responsible in the replication was DNA polymerase, and in the transcription you are having RNA polymerase. Here you are having as like the same, like the direction is 5' to 3'. The catalyzing direction is 5' to 3'. So basically the strand which is having 3' to 5' polarity, it will be forming the 5' to 3'. And the strand which has the 3' to 5' polarity is called as the template strand. Okay, please deal with. And the other strand which is having 5' to 3' polarity will be called as coding strand. So basically, if you want, like I can draw this in front of like you. This is I am having 3', this is I am having 5'. So ultimately here I will be having 3' and here 5'. So basically, like here I will be forming my mRNA molecule here, because this will be like the 5' to 3', and this will be exactly like with this. So this one, like the 3' to 5' strand will be called as the template strand, and this 5' to 3' will be called as the coding strand. So please deal with the case. And this coding strand will be just exactly like to the formed of RNA molecule. The newly formed RNA molecule will be like the coding strand. I repeat, the newly formed RNA molecule will be just like the coding strand. So please deal with only the only difference between the newly formed strand and the coding strand will be that the RNA will be having uracil and the DNA will be having thymine. The RNA will be having uracil and the DNA will be having thymine. Now what is the basic difference between the uracil and the thymine? So the basic difference between the uracil and the thymine is that the deoxygenated thymine is uracil. Deoxygenated thymine is uracil. So please do remember this case as well. This is a very simple phenomena. You don't need to go for like the very deep, very deep study, but still like superficially you do know everything about the transcription now. Thank you guys. Thank you very much.